Thank you, Robin. Thank you, Jean-Pierre, for that very kind introduction. And thank you to the organizers for giving me the opportunity to present our data. I do have a conflict of interest. I'm co-founder of Watchfrog. I set it up in 2005 because I was so concerned about the number of chemicals that we were detecting that interfered with thyroid hormone signaling. Now, we're all endocrinologists in this room, and we all know the importance and the complexity of the interactions between the different endocrine systems and the neuroendocrine systems. And yet, it's worth underlining that the thyroid axis, when we look at TSH, is the only endocrine system that is routinely checked in all newborns at birth. And just in case you've forgotten, this is because without a minimum of thyroid hormone at the right time, a tadpole fails to become a frog and a human baby becomes a cretin. Now, when Jacques Legrand wrote this, I don't think he already knew that it had to be at the right time. Brain development is a one-way street. It's an irreversible process. If you gave thyroid extracts, as they did in the early 1900s, to cretins, they saw that it slightly improved their mental condition, but there were, in some cases, if they were old, it did not. It actually made their conditions worse. In fact, many of us today think we know, because we've known about thyroid hormone for the better part of 100 years. Kendall isolated it famously on Christmas Day in 1912, and he knew it contained iodine, and that's how he managed to isolate it. Iodine was, of course, discovered 200 years ago in 1813 by Gay-Lussac in Paris. So 200 years ago, iodine. Even in the 1850s, the French government sent geologists around France checking the iodine content of the soil and realized that they could correct cretinism and goiter by giving iodized salt. So today we might have been lulled in complacency about what we know about thyroid hormone signaling. But I think it's important to underline that in the last 15 to 20 years, we have learned an enormous amount about different aspects of thyroid hormone signaling. Obviously, today I'm going to be talking about the thyroid disrupting chemicals that are found everywhere. They're ubiquitous, they're different categories, and they're even found in amniotic fluid. The other thing that we tend to forget is that mild iodine deficiency is increasingly prevalent, not only in Europe, but also in the USA, where we used to think that there was a iodine-sufficient population. And, of course, there are many new ideas on thyroid hormone signaling. And I can't give you an overview of them all, but suffice it to say that it's in the last 15 years that we've learned about membrane transporters, MCT8, OATP1C. Very recently, Chris Chatterjee's group has underlined the fact that there are TR-alpha mutations in human populations, often associated with growth delay or intellectual deficiency. Previously, people thought that TR-alpha mutations had to be lethal because nobody could find any. Now we know that they are, perhaps not frequent, but they are found. And we also know that serum levels do not necessarily reflect tissue levels because, thanks to the transporters and the cell-specific nature of transporter expression, plus cell-specific deiodinases activating and inactivating deiodinases, each cell can virtually tailor its needs in thyroid hormone. And my own group has been active in looking at how thyroid hormone controls neural stem cell activity, particularly proliferation and differentiation choices. I'm just going to give you a couple of examples of the importance of some of this data. Mutation of MCT8. I think evolution made a mistake here. In a mouse, there are two transporters on neurons. In humans, there's only one transporter, MCT8, and if that is mutated, you get a very severe phenotype, Allan-Herndon-Dudley syndrome, and the child, unfortunately, has intellectual disability, hypertonia, muscle hypoplasia, and obviously this is due to the fact that the neurons do not mature properly. But MCT8 is also expressed in the placenta, where it shows clear time-dependent expression. I don't think I can use this very well. reflecting probably when and how the fetal thyroid kicks in. And this fits in with a general idea that has hit the popular press now, that this intrauterine period of one's life are absolutely determinant for one's health later. And indeed, the first 9 months of your life can shape the rest of your life in many aspects of disease tendencies, including metabolic disease, cardiovascular disease, but also intellectual development. And I like to say perhaps it's not the first 9 months of your life, it's perhaps the first 4 months of your life, because in the last 15 years, a lot of data has accumulated showing the importance of maternal thyroid hormone levels in early pregnancy. And I think one of the best examples is work that's come out of Robin Peters' team, led by Tim Corrivar. Came out in Lancet Diabetes and Endocrinology early this year, and where they show clearly that an association of maternal thyroid function during early pregnancy, they went up to 18 weeks, and looked at offspring IQ and brain morphology in childhood. The population was about up to 4,000 women, mother-child pairs, and this is just one set of figures. And as you can see, on the left we have the mean child IQ, and as a function of maternal thyroid levels, and you can see that hyperthyroidism, maternal hyperthyroidism, is as detrimental as maternal hypothyroidism. So when Jacques Legrand said at the right time, he could have also added the right amount at the right time. And on the right, you have the percentage of children with an IQ of less than 85, and again you can see this time a U-shaped dose response with again hypothyroidism and hyperthyroidism being equally detrimental. They also looked at MRI scans for about over 600 children, and they found changes in gray to white matter ratio and cortex volume. So this clearly shows that small variations in maternal thyroid hormone status in early pregnancy has significant effects on offspring IQ and brain structure. Now, often I wonder whether the efficiency of T4 therapy in the postnatal period has somewhat lulled us into complacency when it comes to the main topic of my talk, i.e. chemical exposure during pregnancy and in the postnatal period. Here we have some results from Tracy Woodruff, where she looked at the Demeniex study in the States, and she looked for 50 chemicals, and she found that on average, each woman was exposed to at least 33. So an average is 33. Okay. She looked at phthalates and pesticides, heavy metals, polyaromatic hydrocarbons, and some flame retardants. She didn't look at the full spectrum of what we know to be present in the environment. So clearly, the question is, how many of these chemicals can affect the mother's thyroid hormone signaling, and therefore the child's development, the fetal development, before the fetal thyroid kicks in at 18 to 20 weeks of development? And what I find most disquieting is that people from my team have been looking at different data sets and looking at the actual concentrations that can be found of these these chemicals that Woodruff identified. So she didn't look in amniotic fluid, she looked in urine and blood. So what we did then was to go out and look at other data sets and see what the levels of these chemicals were in amniotic fluid where we could find the data. I just want to point out the iodine uptake inhibitors, perchlorate, 10 to the minus 8, nitrate, 10 to the minus 6, thiocyanate, the phthalate, 10 to the minus 6, I'll come to that one later, flame retardants and surfactants, the perfluorinated compounds, mercury and lead. So, okay, these are individual samples, and we don't know quite how broad the exposure to the mixtures is, but certainly the mothers are exposed to these mixtures, and certainly these chemicals pass the placental barrier. So what we've been looking at in the laboratory is how to identify those that are suspected of thyroid hormone disruption. And this is where we go back to our favorite beast, the Xenopus tadpole, and again take advantage of the fact of what Jacques Legrand said: tadpoles fail to become frogs without thyroid hormone, and humans, babies, become cretins. Thyroid hormone has been totally conserved throughout vertebrate evolution. It's exactly the same hormone in fish, as frogs, as reptiles, as birds and mice and humans. And the receptors are highly homologous, as are the deiodinases, etc. So we stuck a GFP reporter into a T3 reporter into a transgenic line, and we studied what happens to thyroid hormone signaling at a stage where they're non-feeding, so they're theoretically an in vitro model for legal points of view, and yet they're metabolically competent. We know that they have the phase 1 and phase 2 enzymes. And the reason we've chosen to take a transcriptional readout is because there are so many levels of potential interactions of EDCs with thyroid hormone signaling. This is a slide, I'm sorry, the credits have disappeared. It should say Mary Gilbert, who works at the EPA in the States. As she underlines, the first level could be negative feedback at the level of the hypothalamus and the pituitary. Second level, thyroid hormone synthesis. We know there are many compounds that interfere with iodine uptake, with TPO, and organification of iodine. Another major factor, distributor proteins or serum binding proteins, TTR, major target. Peripheral deiodinases, mercury interferes, chelates selenium, thereby interfering with deiodinase activity. Theo Visser showed that years ago, before we'd even identified the actual role of selenium in deiodinases. Metabolism in liver and bile. But of course, at the level of the target cell, the transporters, the local deiodinases, and of course, even though evolution has honed the ligand binding pocket of the thyroid hormone receptor to be exceedingly specific, we are now finding that some chemicals can actually fit inside that ligand binding pocket. And of course, the negative outcome or the impact will be on, for instance, on brain development. So we took the most responsive gene that we could find in the tadpole, an early response gene, it's THB-ZIP, and we linked it up to GFP and put it in our little tadpoles, and you can see that at this stage, they're very small and they even fit on a 1-cent coin. And we've set up a screening protocol where we test for 72 hours. We do 72 hours exposure with regular 24-hour renewal, and you can see that the tadpole lights up. We can put them into little 24-well plates, and so we can do automatic plate reading. I like the little slogan is in vivo information with in vitro tools. So what do we actually get? Well, I'm not going to show you all those substances that we've tested, but let's just show a phthalate DHP. In Chinese studies, it's at 0.1, 10 to the minus 6 molar in amniotic fluid. It's used in all sorts of plastics, medical devices, children's toys. It's a really ubiquitous contaminant now. They're trying to phase it out in Europe, and it's being replaced by other things, but we're not quite sure how well it's being phased out. And as you can see, we run the test in 2 conditions. On the left, you have without thyroid hormone, and on the right, you have the same compound tested in the presence of thyroid hormone, 10 to the minus 8, minus 7, and minus 6. And we add this thyroid hormone because it acts as a primer, and it synchronizes the tadpoles and induces the receptors and some of the deiodinases. So we get better results when we prime. It has an agonist type effect. It potentializes T3. And I want to remind you that Tim Korivar's data shows that too much is just as deleterious as too little. We get the same sort of response. Well, it's not quite the same because here we're seeing an inverse U here, an inverted U with PFOS, a fluorinated compound that's used in paints and popcorn packaging and pizza packaging and things like that. So it's a water repellent, and again, we're getting the concentration in amniotic fluid is 10 to the minus 8 molar, and actually at 10 to the minus 8 we don't really see an effect versus T3 signaling, but at 10 to the minus 10 we do, and we get this inverted U-shaped response curve. Not all compounds are active, thank heavens. Here we can see benzophenone 3. It was brought onto the market to replace benzophenone 2 as a UV filter. Benzophenone 2 was a thyroid hormone disruptor. Joseph Curley's group has shown that. And in contrast to BP2, BP3 has no effect on thyroid hormone signaling. So in the team, what we've done is to put together a representative amniotic fluid mixture and expose our tadpoles to this mixture. And here again, we've run the test against T3, and as you can see, there is a dose-dependent increase at 1 and 10 times the amniotic fluid levels. We also ran in this test NH3, which is a thyroid hormone antagonist, and just to show that we have got a thyroid hormone-dependent response. We've also dissected out the brains of these animals after 72 hours of exposure, and we've done quite wide gene screens on the mixture, and I'm only showing you 3 here. But as you can see, we've obviously looked for some genes that have been identified in genetic screens in families where 1 or 2 of the children have been diagnosed with autism. And as you can see, BDNF, brain-derived nerve growth factor, is downregulated significantly by 0.11 and 10 times mixture. Contactin 4, a cell adhesion molecule implicated in neuronal migration, also downregulated. And of course, MECP2, the protein that's associated with Rett's disease and other forms of autism, is also downregulated. Currently, to get a handle on how neuron and glio ratios could be affected by this representative chemical mixture, with the T4 infrastructure in Paris, we've clarified the brains, and we're now counting the oligodendrocytes and the neurons, and with particular focus on these mountain neurons that disappear under the influence of T3 during metamorphosis. We think they could be a good signal for what's going on. Now, so mixtures do affect thyroid hormone signaling in our tadpole screen. David Rao once famously said, he asked a rhetorical question. Suppose that thalidomide, instead of causing the birth of children with missing limbs, had instead reduced their intellectual potential by 10%. Would we be aware? Would we be able to identify its toxic potency, and would it still be on the market? Well, it was a rhetorical question, and unfortunately, I fear that the answer would be yes. You need population-wide studies to see effects on IQ on children. It's no good, one mother going to a doctor and saying, my kid's not doing very well at school. IQ is obviously a Gaussian distribution. So what we have to do, epidemiology. So what does the epidemiology show us? Well, the epidemiology shows an unexplained increase in autumn spectrum disorders in the states where we've got the best data. And even there in the states, it's 11 states that provide data each year. And in 2014 and again in 2016, the incidence was 1 in 16 children, which comes out because autism is much more frequent in boys than girls, 1 in 42 boys. Now, diagnostics methods have changed somewhat, but not since the early 2000s, since the DSM-4 came into play. So we can safely say that since the early 2000s, this increase is not due to diagnostic change. It could be due to increased awareness, it could be due to some changes in insurance that in the States, you get better insurance coverage if your child is declared autistic than if it's just declared with intellectual deficiency. But clearly, even when you take diagnosis and awareness into account, most authorities think that 45% could be due to unknown factors. And clearly we're thinking of gene-environment interactions, gene susceptibility, possibly exacerbated by chemicals in the environment. And I wish to underline that 40% of the children diagnosed with autism spectrum disorder in the States have IQs under 70. And if you look at the number of children that have IQs under 85, i.e. at the limit of intellectual deficiency, according to the state, it can go up to 70%. Now, those are the children diagnosed with autism disorder. But there's a lot of data out there showing that chemical exposure can also reduce IQ. Just let's consider a population of 100 million people, where you've got 6 million gifted and 6 million intellectually disabled. Normal distribution. If you were to have just a 5-point IQ shift to the left, just a 5-point, you would have a 60% decrease in the gifted, go down to 2.4 million, and a major increase in the intellectually disabled children. And don't forget, I've already said it, brain development's a one-way street, so intellectually disabled child is an intellectually disabled adult that's going to need care for the rest of his life. Such IQ losses and more have been documented for iodine deficiency, maternal hypothyroidism, PCBs, lead, mercury, etc., etc. And we know virtually nothing about the additive effects. So, led by Leo Tresandi, a group of us, well, many of us, were involved in different exercises trying to estimate the cost of endocrine disruption in health costs in Europe. And I was involved in the group with Martine Bellinger and Philippe Grandjean and Tom Zola, looking at neurobehavioral deficits, and we considered 2 chemicals that we could get the best exposure data for and the best associations with IQ loss or autism or ADHD. And these were a flame retardant, PBDE, and organophosphate, and we came out with the rather shocking result of 150 billion per annum. Leo took the whole of the studies, whether we looked at male reproductive disorders, obesity, etc., and was able to show that the cost by health effect was largely due, as one might expect, to these neurological impacts, autism, ADHD, and IQ loss, And within that group, it was the pesticides that was the main culprits. This isn't that surprising when you look at some of these pesticide files. In fact, European Food Safety Authority recently published an opinion on 287 pesticides, looking specifically at neurotoxic outcomes and thyroid hormone associations, and they found that 101 of the 287 screened pesticides, when you say screened, they were looking at the files closely, had effects on the thyroid axis. You sort of wonder how they get on the market. So I would like to conclude that over 15 chemical classes are found in most adults and children worldwide. These chemicals pass the placenta and they are found in amniotic fluid. About 2/3 of the chemicals that we have looked at in the lab can disrupt thyroid hormone signaling at some level. And I think this should be put into the context of the increasing number of studies that show the importance of thyroid hormone signal in early fetal development. Other studies show that maternal exposure to certain of these chemicals increases the risk of neurodevelopmental disease or IQ loss. And as I have said a couple of times, we have virtually no data on the combination of cocktail effects, nor do we have data on whether these cocktail effects, these potential cocktail effects, could be exacerbated by iodine lack. And I think it's important to underline today that legislation on the endocrine disrupting properties of pesticides is currently blocked in the European Union by chemical lobbying. And this is unfortunately easy to do. We've seen it for climate change, we've seen it for tobacco. Create, manufacture doubt. And it's given that chemical testing is not good enough at the moment, and unfortunately, we can't really blame the regulators because there's a lot of knowledge that they have to acquire and to incorporate into their data sets. And it's a difficult situation. So with that, I would like to thank my funders and also, obviously, my team. This work is led by Jean-Baptiste Fini in my lab with Sebastian, Michel, Melody, Bilal, and Petra, and the other part of my team works on non-endocrine disrupting subjects. And I thank you for your attention.